Okay, this is the second part of the co-opting the P-element prep material. Where we finished off, we had done a screen. We had a whole bunch of individuals that we had collected that might have the non-autonomous P-element on the chromosome we're interested in, and don't have the wings clipped element anymore. But we don't know which of the flies that we're looking at do and which don't have the non-autonomous P-element, the one we're using to move around and make mutations. And so we're stuck doing still a lot of work, right? And this is not different from using chemical mutagenesis, but we can do better than this, because if non-autonomous elements don't have a transposase gene, but we could engineer them and put something else in there that would allow us to figure out which flies do and which flies do not have a P-element. And then instead of making thousands of crosses with individuals, making those chromosomes homozygous, seeing if there's a phenotype or not, all we have to do is find the flies that have a P-element and then test them for homozygous phenotypes. And it cuts the work down by many fold. So it's a big time saver. So what we use are called reporter genes, and reporter genes are ones that we can use to physically just look at what the fly looks like and see if it has a P-element or not. So if we're engineering the P-element anyway, and there's a number of different interesting features that have been added, which I'm only going to talk about a couple of them because for our purposes that's all we really need to know. But if the transposase gene is removed, you could put something else in its place. And what we do, not we, but the people who did this, they put the wild type cDNA for one of 3 genes in there. These two are eye color. So if a fly is mutant for rosy, it has pink eyes. But if it has rosy plus, it's got wild type red eyes. If a fly is mutant for white, it has white eyes. But if it has the wild type, it has red eyes. Okay. And for body color, so if it's wild type for the yellow gene, it has a tan body color. And if it's mutant, it is yellow. And the yellow is quite striking. Both the white eyes and the yellow body color can be seen through a vial. You do not need a microscope. And this is one of the features of a reporter gene. The phenotypes have to be really easy to see. If they're super subtle and only like 5 experts in the world can reliably every single time tell whether it's wild type for that or not, we don't want that. We want something that even people who are just beginning in the research can look and see, is the P-element present or not, and white eyes versus eyes with some pigment, red, pink, orange, etc. That's a really easy one. Nobody makes a mistake between white eyes and eyes that have pigmentation, right? Some red color. So that's the idea. And so the other thing is, if that's the case, then when you're doing your experiments and your crosses, you use flies that are mutant for these traits. So the flies, if they don't have the P-element, are going to be mutant. Their eyes are going to be white, or their eyes are going to be that pale pink color, or their body is going to be yellow. And then if the P-element is present, the eyes are going to not be white in those flies. They're going to be pink. Or if the flies are mutant for rosy, they're going to have wild type eyes. Or if they're mutant for yellow, they're going to have a wild type body. That's the idea. And so it's almost like putting a little tag or label on the P-element, because if the P-element is present, the wild type gene is present. And as long as you've done your experiments properly and all the flies that you're working with are all homozygous mutant for those traits, then we can use them. And there's standard strains. The commonest ones to use, rosy is still used by lots of people, but the easiest ones are the white and the yellow. And these are both on the X chromosome. So there's a lot of strains that are mutant for both yellow and for white. And we use those as our wild type controls in a way for some kinds of experiments. Okay, and for when we're doing p-element experiments, because then if we see any pigment in the eye, we know there's a p-element present. If it's a white plus p-element, or if we see any wild type body in the cross, then we know that's the fly that has the yellow plus p-element. I hope that's clear. Okay, so for the next slide, I'm going to show you some genotypes, and then I'm going to ask you to tell me what the color of the fly with the p-element is. Okay, so we're looking at the yellow gene in this case, and so just some nomenclature. When we have the lowercase y, that's the mutant allele, and it causes the color to be yellow. And as I think I said this is the second time I've recorded this because I made some silly mistakes and I had to start over. The yellow is really bright yellow. You can just see it through the vial. If you've got wild type and yellow mixed together in a vial, you can easily see which is which without any microscope. It's on the X chromosome. That's not really relevant, but I need to tell you this because there's some possible confusion coming up. The way we designate a wild type allele is lowercase yellow with a plus superscript. Plus means wild type. And then here's the potential confusion. And it's just unfortunate, but the Y chromosome is the Y chromosome, and it's indicated by an uppercase Y. There's no yellow gene on the Y chromosome. It's on the X. Most of the Y chromosome is for making sperm tails and other aspects of fertility for males. There's no sort of essential genes, and so there's no gene for the body color, right? And so when we make a genotype, if we have a male, it's got an X and a Y, and then the way you do the notation is you just put a little superscript. That's the gene, and it's a lowercase. And then if it was wild type, it would be plus, and if it was mutant, it would not have the plus. And so I'm just mentioning this because it's really unfortunate that this is a Y and this is a Y, but just remember that lowercase is the gene and the uppercase is the actual chromosome. Students commonly mistake these because, of course, it is the same letter, but hopefully you will see that, and with a little bit of practice you'll get used to it. Okay, TM6 is the name of a balancer chromosome for chromosome 3. T means third, the third chromosome. M is multiple for multiply inverted, and 6 is just the sixth one that they designed. These were made deliberately and then isolated and tested, and the ones that were successful have been named TM1 through 6 or 7. There's a number of them. TM6 and TM3 are the most commonly used ones, but I know there are some others. TM1 and TM2 also. So here's our situation. We've got a p-element insert on chromosome 3, and we want to make a balanced stock. So this is the aim. What we want to have is we want to have a stock that everybody's homozygous or hemizygous, mutant for yellow. And then on the third chromosome, they've got the mutant, the PIA insert, and a balancer. Okay, so we want males and females to be these genotypes. Okay, and we've just finished our experiment, and we've isolated, in this case, the way we did it, we ended up with females, and the females had this genotype. So we know that they've got the P element, and now what we want to do is we want to make males and females that have the P element as well as the mutant background, right? And this is what we have to cross it to, because that's the stock that's available to us. Okay, plus just means there's no P element, no mutation on this allele. So in the next generation, we've got 2 different genotypes that we're going to see. Female progeny, and we'll have male progeny too, but this is just for collecting the females. And I just want you to think about, I hope this will be clear and easy to do. Just put your answer here about which of these will it be, this female, or this female that we're going to collect in order to make a stock, which will give us, because we'll also get males that have this, a Y chromosome, and this constitution, so that we can make this stock here, okay? And I'll be asking that in class. So if you think of it in advance, your learning will be better because you will have figured it out, and then you'll be able to find out if you're correct. If you write down what I say in class, you will know the answer. That's not the same as learning, okay? Just so that you know. Okay, so reporter genes are one component, and I would say the most important aspect of or component of engineered P elements. Engineered P elements are ones that we've deliberately made, and they have features added to them to make our life easier when we're using them to induce mutations. And then the first most important thing is being able to always know which flies have your P element insert and which don't. In the same way as, you know, having a dominant mutation on a chromosome lets us know that any fly that has that mutation, that phenotype, has that particular genotype. Okay, everything we do is designed as much as possible so that we can just look at them and know what their genotype is. Okay, for the most part. Okay, and so the way we do this, you know, this was done genetic engineering with genetic engineering. The transposase gene was removed from a wild type element, and the reporter gene was cloned in its place. The whole element is added to a plasmid, which is a circular piece of DNA, which has some features that are required to grow in bacteria and so on, but will have also had our P element with its terminal repeats at each end added to it. And then they are introduced artificially into flies. We inject them into early embryos, and then we select, and we use the reporter gene, the white plus expression or the yellow plus or the rosy plus, to collect the individuals that have the P element. And there's a little complexity to it, which is why I'm not including it here as part of your learning. If you were in 357 last semester, you got a bit of that already. If you want more information, though, there is a recording that explains it. This is optional, okay? Some students like that extra information. It makes them feel more confident and more comfortable about understanding the topic, and for some students, they don't need that to feel comfortable. So it's there for you if you want it, but you don't have to. I'm never going to test you on that, that knowledge, any more than what is just here. We introduce them through injection, and then we have to do selection to find them. And the reporter genes are essential in us being able to find the flies that have the element. And then once they're introduced, then we have a strain that has that. Those are starting material for the kind of screen that I talked about. So we've got a fly strain that has a P element in some position on chromosome 2, and we're going to use that as our starting material, and we're going to make mutations on chromosome 3 in the kind of screening process I've already just gone through. So that's the kind of thing. That's as much as you need to know. Okay, so now many genes are known only by mutant phenotypes. Not as many as before, but we've had the full, almost the full genome of fruit flies sequenced for 25 years or so, something like that, 26 maybe. And still we don't always know for some strains what mutant phenotype we're seeing, which gene it's associated with. So knowing the DNA sequence isn't good enough. We also need to demonstrate that this particular gene out of the 8,000 on this chromosome are the ones that are responsible for this particular phenotype. So we don't, you know, you would think once you had a full, people used to say, oh, well, once the genome is completely sequenced, there won't be anything else to do, but there really is. There's lots to do still, because we need to know more about how those things work. And if we've done chemical mutagenesis, which a lot of our older mutations are, it's not like you can just look at flies and go, oh, that must be the blah, blah, blah gene. Sometimes it takes years of work to actually figure out what the gene is. And so the value of a P element induced mutation it's twofold. In one case, it's reversible. You can take the P element back out and go back to wild type, which is very interesting and very valuable. And I'll talk about that in the next recording. But I'll also talk about how we can use what we know about our P element sequence to figure out where the P element has inserted. And I'll just start you off with this slide. Now, if you don't have any or very much background in PCR, you may find this a little bit confusing, but I will try to explain it simply. And then after, in a couple of weeks, we're going to have a whole lecture that's all about PCR, both how it works and how we do controls and that sort of thing. And if you've been in 357, you already know how it works, plus all kinds of fancy extra things you can do with PCR. But we'll start off with this. So basically what you have is you have a P element that's inserted into DNA, either right beside or right inside a gene. The blue arrows here are the inverted repeats. And when we're using an element like this, we know the entire sequence, including restriction enzyme sites, which I'll talk about in the next recording. And so the value of using insertional mutagenesis is that we know some of the DNA sequence of the element we used, and we can use our knowledge of this DNA to find out what the DNA next door actually is. And so give that some thought, and then in the next recording I will talk about that and about reversion testing.